Hello, everyone. Welcome to the 19th Antibodies Journal Club. And today, joining us is Dr. Ian Myles. He is a tenure track researcher at the NIH in Bethesda, Maryland. And his main research focuses on using strain of bacteria collected from healthy people as a topical treatment for eczema, as well as examining the environmental exposures that influence human health through impacts on microbiome. Um, Ian, welcome to the Journal Club. We're really glad to have you here today.
Thank you for inviting me.
So, you mentioned that you mostly work on impact of bacteria on eczema. So, how did you come about working on this research with breast milk and the immunity?
Yeah. So I got into this from med school. We'll talk about it in the slides, but I heard about it before I knew enough about immunology to try to dismiss it. So there's something in the past called transfer factor, and I heard about it as a med student, and I wanted to look into it. So when I came to the NIH, it was something that was on my list of projects that I wanted to do. And then when I took a job in the lab, it was focused on basically staph skin infections and staph predispositions, which as an allergist, most of the staph patients are going to be your atopic derm folks. You'd struggle to say these two things are directly related, but they're just parallel projects. So this has always been something that I was intrigued by, the science of it.
Okay. And last thing before we move on to the presentation, you mentioned that your 5-year-old boy is obsessed with superheroes and he likes Marvel more than DC. So,
Like every normal person should, right?
so it looks like you're also a Marvel person.
Yeah, I guess. I mean, growing up, I was Batman, I suppose. But so he's only 5, so he can't watch any of them because it's too violent and they use bad words. But it's like I've made the joke. It's like the Greek oral tradition. Like he's heard the entire cinematic universe story through asking me questions about, like, well, why did Falcon have this wing? What happened? Where's Winter Soldier? Why did Captain America go to jail? Like he could tell you the whole history having not heard it. So he's obsessed. Um, it's fun.
Yeah, I'm also in the Marvel bandwagon.
Yeah, now he's into Miles Morales, right? 'Cause my last name's Miles, and
Oh,
you
so
know the
it
new
just
Spider-Man.
I didn't want to bring it up, but oftentimes when I heard of your name, in my head it would be Miles Morales, and I'm like, no, that's that's not what he's named.
It is in my son's head. That's what his name means now. He's the new Spider-Man.
All right, so with that, let's begin our session today. Ian, I'm going to transfer the controls to you. Mike is all yours.
Okay. All right, that's working.
Yeah.
Okay. All right. So first one, we're going to throw out a question. Everybody know what melatonin is and what it does? Somebody can answer this. Go
Is that
for
the
it.
sleep hormone?
Yeah, that's right. It's the sleep hormone that tracks with your circadian rhythm. So this is all the fancy gadgets that you have nowadays that try to switch to blue light and red light depending on the time. The theory behind it is the induction of melatonin in the brain. So if you track the melatonin concentration in a woman's breast milk throughout the day, this is what you get, right? You get this big spike towards the evening. And I think we can all, if I said, why do you think that is? I think we can all piece together that it's going to be some kind of mechanism to get that kid to fall asleep and stay asleep. I'm sure you're all too young to have children. One day you will truly appreciate the quality of this book, and you will enjoy it. So you understand though immediately why melatonin would spike in the evening for breast milk. How about this? So this is from cows. Most of the milk research is in dairy cows for obvious reasons, but this is true of every mammal that anybody has ever looked at. All right. So if you look here, this is the percent of CD4 and CD8 cells in the milk of the cow as it's going along. So this is during the cow's pregnancy. And this is, round about the CD4, CD8 ratio you would anticipate. This line is parturition. So this is where the cow gives birth. And you see this immediate flip where suddenly there's a selective enhancement, enrichment, I should say, of the CD8 cells in the milk, particularly early on. And that tends to fade out. We don't know much beyond a year. But again, this is true of every mammal ever tested. And then in higher level studies, it's not just CD8 T cells, it's effector memory CD8 T cells. And we won't have, in 30 minutes, we can't go through all the nuances between them, but You get the idea. It's not, I mean, it is entirely effective memory CD8 T cells early on in mammalian features. So why is that? I don't think that that is so obvious. You're taught that CD8 cells are not helper cells. You're taught that these are maternal cells, so therefore shouldn't have any capacity to do anything in the offspring. And this is true for our own work. So what we did here is the number of CD8 T cells per milliliter in the blood from 8 different women at 10 different collection time points at their postpartum dates. So the blood, you can see it's fairly stable, which you would anticipate in the milk. There is some variability, but it's always an enrichment over the blood. You look at the CD4 to CD8 ratio. Again, the red line here is the blood average, not a whole lot of variability, but early on you have a selective enrichment for CD8 T cells. Subsequent analysis shows that later on you get CD4 T cells. There are other groups that are working on why the CD4 cells are there and what they might be doing, but they're not going to be relevant to what we're discussing. And then once again, in terms of, it's not just CD8 T cells, we find the same thing, the effector memory to central memory ratio. So this blue dotted line is the average effector to central memory ratio in the blood. And then the blue dots are the the milk sample cells. What are those ratios? And so once again, an enrichment for effector memory T cells and an enrichment for central memory CD4 T cells. Again, I don't know how to explain the central memory CD4 cells. We're only going to talk about the effector memory CD8. Okay. So we got these weird cells. They're showing up in breast milk. Why on earth might that be? So to do this, we're going to step, when I say back, we're going to go way back to the 1800s, where Robert Koch, somebody can correct my pronunciation, but this is the same Koch's postulates guy. So he discovers and isolates tuberculin as one of the central antigens that stimulate your tuberculosis test. That's what gets injected in the skin. Charles Manteau discovers that you could use this as a test for tuberculosis. 40 years after that, somebody named Merrill Chase comes up with the concept of cellular immunity. He won the Nobel Prize for this. This is taking cells out of the peritoneal cavity of guinea pigs and showing you could transfer those cells to another guinea pig, an immunized guinea pig to a naive guinea pig, and you can transfer immunity. Takes for granted now, but this was a big deal obviously back then. So like you do in any studies, right? You discover something and then you go through and try to break it, right? Like, what is it dependent on? If I block this, if I block that. So the guy who worked in the office next to him, H.S. Lawrence, he wanted to see what might be happening. So he takes the peripheral cells from humans, or he worked on mice, not guinea pigs, and then basically froze them and thawed them so many times that the cell integrity was lost. And can you break it up into pieces? And will you still get some evidence of transfer? And so what he would do is DTH stands for delayed type hypersensitivity. This is the concept of the tuberculosis test, right? They do an injection and they look a few days later. So you find somebody who's got a positive TB test, you pull out their white blood cells, you put them through, you freeze and thaw in water, and then it'll break the cells into all the different pieces. And then he would dialyze them to try to say, like, all right, what size molecule do I need to get this transfer? And then he would take this dialysis and inject it. So just burn this cartoon picture in your head for if you're watching this online. This is the cartoon we're going to use if I say a cell lysate. So I'm going to take that cell lysate, I'm going to inject it into something. That's how I'm depicting it. And then that person would suddenly develop delayed type hypersensitivity to, say, tuberculosis, even if they were negative beforehand. And it's important. If the source material was from somebody who wasn't positive to tuberculosis, then the outcome would not transfer. You do not generate responsiveness if the host material, the starting material, wasn't from somebody immunized. It also wouldn't work if there was a mismatch. So if you took somebody who was PPD positive but wasn't diphtheria positive, you're not transferring diphtheria reactivity, but you can transfer tuberculous activity. So they took this even further in mixing studies. So they took 2 groups of donors. One was diphtheria positive, tuberculosis negative, then diphtheria negative, tuberculin positive, right? And you took you split up 3 different groups of recipients. Both were skin test negative to both of these antigens at the same time. If you give whatever you gave the diphtheria positive versus you gave one group just tuberculin positive, and then the other group got a mixture of both. So suddenly the group that only got diphtheria was positive to diphtheria. If you only got tuberculin, you were positive to tuberculin. If you got both, you were positive to both. All right, so this is really starting to hammer home the idea of antigen specificity. Subsequent research used novel antigens. You guys know of like KLH or some of these things that aren't ubiquitous in the world, so that you can't worry that maybe I skin tested the person and then they became positive because I skin tested them previously, even if they were negative for that skin test. This is just to say no one in the world is allergic to this. So if I test them to this, they're basically hammering home the idea of antigen specificity. There are big problems. Remember, this is 1950, so lots of difference in the world. Nobody had any concept of a molecule this small having any immunologic effect. The idea, like IL-1 beta was just barely discovered. I mean, they weren't even at the IL-2 level of numbering yet. So no one really fully appreciated this. So now you got somebody who's coming along saying this is an incredibly small molecule, and it has antigen specificity. All right, so what's the problem with that? So everybody knew about antibodies having antigen specificity, and I'm just show you the relative size. So if we set antibodies to Shaquille O'Neal, the T cell receptor would be the only other thing in immunology described as having antigen specificity. That would be the size of Bo Jackson, the football player. And if you're talking about something as small as what this transfer factor or dialysable leukocyte esterase, it's the size of an adorable 2-year-old boy, right? So like nobody could say to you that it didn't make sense to say, well, how could something this small carry antigen specificity when the only two other things described are far larger, and there's nothing even in immunology at that point that was that small. So the other problems with this work at the time the first one was donor inconsistency. So when I told you I take something from somebody who had tuberculin positive, for example, it wasn't universal that that person would be able to transfer, right? So the positive predictive value of that tuberculin test, which means if I take 10 people and all 10 of them are tuberculin positive, it's not that all 10 of their cells will transfer reactivity to a naive person. It's never 100%. But now, if you have people who are not tuberculin, like 10 negative people, 0 will ever transfer. But it just said that having a skin test positivity wasn't enough to predict whether or not this person was going to transfer immunity. So that was inconsistent, and that was never explained. At the era, you started to have a lot of people who were a little bit Looney Tunes, we'll just say, making a lot of different non-immunologic claims. They seem a little less Looney Tunes now because I think the concept of saying Alzheimer's might have an inflammatory component, not controversial to say today, certainly would have been problematic to say in the 50s. You could even say something like autism has an inflammatory component today and get away with it. Not so much then. So it puts people on the wrong foot. And then you had a booster discrepancy. And this was the biggest problem in the 50s and 60s for why this research didn't move much further. So I told you, you take a mouse, you immunize the mouse, you can pull out the spleens and you can inject it into the new mouse and you get reactivity in the new mouse. Merrill Chase attempted to recreate this using guinea pigs. And what he did, however, was immunize multiple times because that's the protocol that they used at that era, and it failed reproducibility. So now what you have is you have a Nobel Prize winning lab telling you it doesn't work. And then you have a handful of people who are associated with some lunatics telling you we think it does, and you can get the sense of where that research went forward, right? So that just then became relegated to a handful of very small labs. We're going to come back to I'm going to ignore the non-immunologic claims, that's beyond my scope, but we'll come back to the donor inconsistency and the booster problem in a bit. So I'm going to show you our animal models, but just give you the general idea. You immunize a mouse with complete Freud's adjuvant, which is like a dead tuberculosis, and it generate whatever you mix in there, you're going to get a profound immunologic response. You give it 3 weeks, and then you can do a foot pad test, and the mouse foot swells up. That's how you do a TB test, for example, in a mouse. Same deal. Pull out the splenocytes, put them through freeze and thaw, dialyze the cells down, inject it into the new mouse, and then after about 24 hours or so, you can challenge that mouse to the antigen and you get foot pad sensitivity. All right, so the first thing we did is we wanted to see is any of this, go back to the antigen specificity. So we take this mouse We're going to inject it with splenocytes from mice that have been immunized to ova or immunized to BSA. Okay, so bovine serum albumin or chicken albumin. And if we just take spleens that we didn't immunize the mouse at all, so you just take spleen lysates basically for non-immune mice, you don't get any difference in the foot pad swelling induction in the naive mice. If you take mice that you injected with just CFA to see if there's just some non-specific immune activation that you're generating in the spleen and then transferring to the mice, you don't get any difference whatsoever. But if you had cell lysates that came from mice that were immunized with ova, you don't get any response to bovine albumin, but you do get specific foot pad swelling to chicken albumin. So you are transferring ova sensitivity, but not just albumin sensitivity. So some evidence of antigen specific in vivo. That, this setup was basically what they had done in the mouse world. And to their, in their defense, cytokine biology, all this kind of stuff just wasn't a thing. So what we did next was we wanted to see, okay, do we have to use a foot pad test, or can we actually pick out a cytokine response? So once again, we inject a mouse with splenocytes or cell lysates from an ova immunized mouse, take the splenocytes out, and then put it with an antigen pulsed dendritic cell. And we looked at multiple different cytokines. The one that came up, always consistency, was IL-17, particularly IL-17A or F. Again, IL-17 wasn't known entity back then when they started, so there's no way they could have found it. So if you take the dendritic cell and again, you pulse it with bovine ova, you don't get any, or bovine albumin, you don't get any response. But if you pulse the dendritic cell with ova albumin, you get a very strong IL-17 response. The next question was, do I have to inject it into the mouse, or is it doing something directly on the cells? And so now we just take that lysate and throw it into the culture. And in both a time and a dose-dependent fashion, you get this induction of IL-17 directly out of the cells. Importantly, though, and we'll come back to this, you needed both cell types. So we're not inducing IL-17 out of dendritic cells. We're not directly inducing IL-17 out of the splenocytes. You needed both of those cells in there. So there's something happening in the interaction between. Other things that we could start messing with, right? So this is our basic mouse setup. So just like anybody would do, you could use different knockout mice, right? So you're basically going to answer the question, which pathways are essential to have this response? If I inject a knockout mouse with this lysate, can I lose that IL-17 induction activity? Obviously, an IL-17 knockout mouse would, but what about everything else? You can start to pre-filter the cells. So rather than just taking spleen lysates, let's just take lysates from CD4 cells or CD8 cells or CD19 cells, and you can say which cells are essential for that lysate function. You can mix and match the spleen, the splenocytes again. So you can say, all right, well, which cells are actually producing the IL-17? And then you can just, again, independently do splenocytes and dendritic cells and say which cell is the actual target for this spleen lysate activity. And so when we started mixing and matching some of those, you can see the IL-17 induction. If I run it through a CD8 depletion column before I make the lysate, that lysate does not induce IL-17. And then the inverse, where I clean the column out and basically pull the CD8 enriched fractions, you get an IL-17 induction. And that's the same thing with the foot pad swelling. So if I sort out the CD8 cells and just use that CD8 fraction, I get almost the same induction as if I just didn't sort them at all. But if I only use CD4 cells, I don't get that response. And then if I look at the dendritic cells, and I'm focusing now on IL-6 or the combination of both for IL-17, we use something called tetramers. So tetramers, I assume most people in the audience know it, but it's a flow cytometry thing with 4 different antibodies, all matched, and then it has a fluorochrome on it. So for this one, it's the you're actually using a T cell receptor type fluorochrome. So what I'm trying to find is I want to look for the dendritic cells that I'm looking for the splenocytes that react to ova. So I'm pulling out the very specific ova reacting T cells, not just CD8 cells. I just want the cells that only are reacting to ova. And if I use those lysates, I get response to dendritic cells, and I get the IL-17 response in the complete package of cells. And if I use even a CD8 positive tetramer negative cell, I get an inhibition of IL-17. So this is now arguing for very specific antigen specificity. And with tetramers, you're now getting to epitope specificity. All right, so then just a quick word about the T cell receptor. I told you, it's the size of Bo Jackson in our relative world here, but that part that actually is specific to the antigen is incredibly small. You're talking about 10 to 20 amino acids. This is the CDR3 hypervariable region, right? The rest of it is a structural compound, so you wouldn't anticipate that would be transferability, because that should be host specific. But in terms of antigen specificity, you're talking about a very small subsection of the T cell receptor. And so they make a mouse that already has the OVA responsiveness built into it. So it's an OT1 and OT2 transgenic mouse. And so what they do is they genetically insert either the CD8 epitope or the CD4 epitope. So now every single T cell will have a TCR on it that responds to either the CD8 or the CD4 epitope. And so if you use a dendritic cell and you pulse with OVA, so in these mice, I don't even have to immunize. I'm not doing anything. I'm just ordering them from Jackson, and I'm taking their splenocytes and I'm dialyzing them. And then I'm throwing that lysate into antigen pulse dendritic cells to see what happens. So again, if I take the OVA immunized mice, so this is my baseline stuff, I can get a pretty decent amount of IL-6, but a massive more if I take the OT1 lysate. But I don't get anything if I take a lysate from an OT2 mouse. And it's specific because if I use heat-killed Candida, I don't get any response. And I can even go down to the level of the epitope. These are the individual amino acids, the synfecal protein that you stimulate the dendritic cells to. And again, you get an IL-6 response specific to the OT1 lysate. So you're matching those two antigens and epitopes. And then further evidence of TCR involvement is, so now I took the splenocytes and I'm going to antibody deplete things. So now I'm going to try to pull down TCR beta out of my lysate before I do subsequent studies. And so if I pull down TCR beta, I can basically deplete out all the IL-6 induction out of the dendritic cells. If I elute it, I can get it right back. And the isotype control is the opposite. So I'm now able to show you I can pull the reactivity out with a TCR beta antibody. So this, to some extent, might start to address the donor inconsistency. Now remember, the tuberculosis test in your head is a CD4 reactivity test. It's not necessarily a CD8 test. It was built to pick up DTH, which is CD4 reactivity. So now we're going to take a patient, an individual from a different host. So these are two individuals, both of whom are skin test positive to Candida, right? Everybody in the world should be skin test positive to Candida. It's a screening tool to see whether your immune system is functional. If you're skin test negative to Candida, please go see a doctor. All right, so this person is just a healthy volunteer. And in this cell, we take all their cells, and these are the CD8 T cells, and then this is the CFSC. So you're stimulating these cells with now Candida. And as a cell divides, it's going to move this way. And so what that means is it's going to dilute out the dye each time it divides. So what you're seeing here is this cell over here is highly reactive to Candida because it represents that it proliferated a bunch in response to Candida. And you'll notice that and this person is all CD4, right? Because it's CD8 negative, it's CD3 positive. So at least it's CD8 negative. They don't really have any CD8 positive cells reacting to Candida to speak of. This person, which is labeled Ed, is an enriched donor. So this individual is a healthy person whose wife and mother-in-law both live with them, and they have specific genetic disorders that predispose them to Candida. So the wife and the mom are always getting sick with Candida. So my hypothesis was basically, if anybody in the world has got an over-exuberant exposure to Candida, it's a guy who lives with 2 Candida-infected patients, and let's see what he has. And so he's got plenty of CD8 highly reactive cells in his peripheral blood. He's got CD4 as well. So if you took the skin test for both of these, they're both positive, but only one of them has actual CD8 responsiveness. And so now when I do the same thing, I'm going to take the lysates from these different individuals. We did multiple healthy volunteers. And so the healthy volunteer does not induce any IL-6 out of the dendritic cells pulsed with Candida. But the enriched donor does. And I think that could potentially explain that donor inconsistency, because the skin test is not an assessment of who has CD8 reactivity, it's just who has any cellular reactivity, particularly CD4.
Were these dendritic cells taken from the same donors for
From
testing?
within an experiment,
Yeah,
yes.
yeah. Okay.
So
So
like
it's
we went to the
matched.
blood. What's that?
So it's donor matched, the dendritic cells and the lysates.
No, no.
Oh.
The donors are consistent across the experiment, but they're
Mhm.
not from any of the specific individuals.
Okay.
So this is not you do not need to match the individual. So I don't even know what the HLA types is. So you don't have to match those things.
Okay.
And now we're going to talk about that fight back in the day between the guinea pigs and the mice. So I told you that the guinea pig protocol actually immunized 4 different times. Because if you want to induce a strong, robust CD4 response, that's what you do, right? Everybody does booster shots in order to generate these kind of responsiveness. Because if you're focused on CD4, that's how it works. Sadly, even though these two people were beefing with each other, they never actually compared each other's protocols side by side. No one ever actually, let's do one injection versus multiple and see if we can hash out the difference. It was, hey, the Nobel Prize guy said it doesn't work. This other Looney Tune says it does. We're going to blow this guy off. So now we're doing the same thing. Now, these are all mice. But this is just that single injection in CFA to boost up that response. And then this is just one booster shot. And with as little as one booster shot, I can obliterate the IL-17 reactivity. Oh, I mentioned that. So the reason why I think we're doing that, we have some evidence for this showing, is that those booster shots change whether or not you have an effector versus a central memory CD8 T cell response. And so we sorted out the effector memory cells as well as when we were playing around with that, and we could find that this IL-17, IL-6 induction is effector memory specific. So just to show the summary cartoon of that work before we get back to the milk. So you start with an antigen specific and sometimes epitope specific CD8 T cell. It's releasing the TCR beta, some portion of it, unlikely to be the entire thing because that would be too large and that would limit you between mixing and matching cells from different people. I'm not showing it, but we think there's some element of S100, A9, and maybe some other tag-ons, but there's something that's going with it. And it does need to be broken up, right? And so I don't know why yet, but the cells do not release this naturally. It's not like a B cell releasing its receptor for an antibody. Something is, you have to forcibly remove that. That works directly on dendritic cells in both mice and in humans. Things that I'm not showing you, it's dependent on MHC1 expression. There was some element for TLR4. And then the inductions are kind of your standard pro-inflammatory pathways, nothing unique. And then that IL-6 turns around and works on memory CD8 T cells to again induce IL-17 and to some extent a TNF alpha response in the host. So
Ian, can I ask something here?
Mm-hmm.
So it looks like just free-floating T cell receptor beta is inducing a response in these dendritic cells, right?
So this is just my cartoon. We got a very expensive mass spec device being installed next week, and I will reserve the right to redraw my cartoon when we get the data to back it up. But what I think is happening is that there's got to be some assembly of, it doesn't have to be the whole TCR beta. Like I said, if it's just a chunk that include the hypervariable region, that would bind to your antigen. But then there does have to be something else stuck to it that is driving the response. Because we can order just recombinant TCR beta and we don't get it. So something else is in that cell that's tagging onto it. But yeah, I think my hypothesis is that the TCR beta or some chunk of it is coming up and interfacing with the dendritic cell, and then some other portion of it is interfacing with those other receptors. And it's hard because spatially we still think of it like a cartoon, but these cells are completely folded on each other all the time. So it's not unreasonable for one cell to activate receptors on different parts of the cell surface. So that's what I think is going on. Okay, so that's how we came to it. We came to it as hey, this is a story about some weird people who used to work on these cell lysates. I'll tell you, I came on, and I'm not that old, but after med school, it was enough where it had already died. And what really killed that research, other than the fact that you had a Nobel Prize lab disagreeing with it, was HIV and hepatitis C, right? So taking cells out of somebody, deriving a therapy out of that blood and putting it back in the 80s became a very stupid idea because you just didn't have the HIV hepatitis screening precautions in place. And you would only do it if someone needed a blood transfusion because they're otherwise going to die. So that basically killed that. And so we wanted to come back. And so what that basically meant was when we look to see, hey, what are the cells that are able to passively inhibit or inherit cellular immunity? And if you say, well, what's the only physiologic condition in which passive immunity would ever exist in terms of nature? And the two situations from the breast milk and our work kept pointing back to T effector memory CD8 T cells. All right, so now we jump back to the milk. And I will admit, when I started my work, you can go online and you can type in the word transfer factor and it'll come up with all kinds of dietary supplements. It'll say it's from from colostrum and whatever. And when I started, I thought these people were the lunatics, right? Like, come on, there's not going to be an overlap between these two things. But there is some evidence that the cells in the milk mean something. So if you take two different sets of women, both of whom are PPD positive, versus a moms who are PPD negative, right? And so you're going to separate the PPD positive, whether they are breastfeeding their kid or bottle feeding their kid without breast milk, right? Formula feeding. And so they take the cells out of the infant, typically the umbilical cells, right? You're not doing a blood draw for this. And you take the umbilical cells and you say, okay, we'll stimulate them with PPD. Now, this is an old study, so they didn't have all the fancy stuff, but they're just looking for, again, cell proliferation. So if the mom is PPD positive and she's breastfeeding, the offspring cells will react to tuberculin. But if the mom is PPD positive and she's bottle feeding, the baby cells will not. So there's something that's happening, again, transferring in the milk. Now, they didn't sort through this, is it CD4, CD8, or what have you. But again, this is some evidence that those cells matter. And so we did something kind of similar with cross fostering. So you take wild type mice, and so as they're born just regular, and then they're going to feed or nurse with either wild type, also kind of match wild type mice, or mice that are immunized to ova prior to their pregnancy. So when they are ready to nurse, and then or CD8 knockout mice or mu-MT mice, which are mice that don't make antibodies, because as was mentioned in the preamble to this, everything that tells you, oh, breast milk transfers immunity, it is all thought of as an antibody-mediated phenomenon. So you get some induction of IL-6 out of dendritic cells when you're, out of the splenocytes that are re-stimulated with ova from the nursing mice, if you're using an immunized mouse, right? So the mom being immunized shows an advantage to the offspring's ova reactivity. But when you look at the CD8 knockout mice, that's obviously lost. And in fact, if you immunize the mouse, you can actually make it worse, but there's no loss of that transfer effect when you take out the antibodies. So I'm sure the antibodies are doing other things, but they're not doing this specific mechanism. So then we wanted to try this with both human and cow milk. And funny enough, in the state of Maryland, it's illegal to sell colostrum, but they can give it away. So there you go. So we went up to some farm and they said, like, yeah, sure, here, you can have some. And so this is bovine colostrum, and then two different samples at this point of human breast milk, either from day 8 postpartum or day 90. And so this, we were looking at ova or he killed Candida, again, under the assumption that both the cow and the human would be responsive to Candida. And so the he killed Candida induces more IL-6 out of human dendritic cells than it does when you're challenging with ova. And the same thing, it's lost a lot from day 90, but you do still see some levels of antigen specificity. But again, there's more background stimulation and non-specific stimulation from the milk samples, as evidenced by the fact that they react to a dendritic cell with ova. And then the same thing, if I take the cells on my screen, hold on. On my screen, everybody's face is blocking this. There you go. All right, so so if you take the cells, that's right, and you deplete out if you take the breast milk sample beforehand, and I spin down and I take all the cells out of it, but I'm leaving everything else in there, I lose that IL-6 processing. But if I use a column to deplete out the IgG, and we did it as well with IgA, you don't lose this particular phenomenon. And so then we did the same thing with those human breast milk samples where we took the milk and we antibody depleted it for TCR beta. So this is TCR depleted, TCR eluted, and then this is the isotype depleted and eluted. Split those into the dendritic cells. And again, you get more background evidence, but you still find some reactivity. So you take the milk samples and you deplete out TCR beta, you lose some of that activity, and you can elute it back into your media. And same thing, isotype control, you get the opposite. We also, at least in the United States, at a particular time point, every woman is expected to get a booster shot to CMV to see whether or not they are immunized. But we're looking here to see who is CMV positive versus CMV negative by IgM and IgG. So this is prior to the booster shots. And then the PP65 is a particular epitope for CMV that you can buy recombinant and throw into the dendritic cells. And so in the moms who were CMV positive, if we take the breast milk from them and throw it onto those dendritic cells, you get this induction of IL-6, but not the breast milk from moms who are CMV negative. And you don't get you get non-specific activation when there's Candida. I will tell you that the heat kill Candida will activate the dendritic cells more just by its lonesome, but you don't get this induction or any difference of induction of the breast milk based on CMV when you're not using CMV as the stimulant. Same deal with the dendritic cells, where we're throwing either the transfer factor from the cells, or at this point, dialyzed milk samples. And now we were looking for specific genes that came up on kind of our full transcriptomics work before. And so for the most part, you can see consistencies in terms of IL-1 alpha, IL-6, LOX, PGE2, and so forth, that the enhanced donor, the one that from that guy who had a lot of CD8 cells to Candida, the bovine samples, day 3, day 8, day 90, same deal, whether you're putting the dendritic cells on mouse or taking the dendritic cells from mice or from humans, these samples induce very similar genetic pathways in both mice and humans. Now, they're not a unique magical genetic pathway. I mean, it's just the pathway of normal inflammation for a dendritic cell, but the overall consistency is at least intriguing from our end. To see if we could make any difference in the mouse models, again, we took either the human cell lysate from that enriched donor or just dialyzed colostrum. And in this one, we're doing foot pad thickness. So you take a mouse, you inject it with either the human cell lysate or milk. And then we foot pad test to Candida. You can see a foot pad test response to basically equivalent between the enriched donor and bovine colostrum. And in this one, also under the assumption that people should have some baseline reactivity to Staph aureus, when we inject colostrum into the mouse and then come back and just infect the mouse with staph. So at time 0, we're injecting Staph aureus into the skin, and then you're basically watching how big of a lesion it gets, so how big of a wound. And so there's some protectivity from the colostrum. And so wrapping up, connecting the two dots. So, I'm showing you the cartoon for what happened with our cellular work. And now we're finding out that, I shouldn't say finding out, we're realizing that previous people have already discovered that the breast milk is enriched and milk is enriched for antigen specific T effector memory cells. They induce a very similar genetic pathways in dendritic cells. There's partially dependent on TCR beta, again, activating the same pathways. So again, a lot of things pointing to the potential here that we joked about in the beginning. Why is melatonin in there? So we know why IgA is in there in terms of giving the offspring a better humoral immune system during breastfeeding. So why might those cells be in there? Well, it might be to, again, provide a temporary transfer of immunity. And so those, they're enriched in early breast milk samples, seem to transfer immunity both in vitro and at least in mouse models. It could represent passive transfer of immunity, which would make sense as to why those maternal cells would be in there at that level. And again, what we need to do is mass spec analysis of, we're going to focus on the mice and look for the OT1 lysate versus OT2, since those should otherwise be almost identical, and see if we can pull out a signal for a molecular signal of exactly what transfer factor is. And then we'll go hunting through breast milk to see if we can find it, because obviously breast milk is far more complex and would be hard to find. And so with that, open for questions.
Thanks a lot, Ian. So I got several things in my head.
Okay.
I'll just start with one and I'll let others ask if they have anything. So in the experimental model, we had the cells that were lysed in freeze-thaw conditions, right?
Mm-hmm.
But in the breast milk, how do you think the lysed cells are coming over there?
Yeah, so we're in our hands, we frozen thawed the breast milk, and then did the same thing where we put it through the dialysis.
Okay.
I'll warn you, you have to kind of spin a little bit out, because otherwise the lipid content for colostrum is astronomical. So you spin it out and you get like this layer of fat, and then you can freeze and thaw the rest of it. But in the gut, if that baby ingested those cells, they will lyse very quickly. So in studies where they've taken mice and fed them, basically just fed them splenocytes, the neutrophils and the macrophages will make it out the other end in the mouse poop alive, right? So maternal neutrophils and macrophages can survive the passage. And you would have to presume innate cells should do innate things on their way through. The CD4, CD8 cells do not. None of them make it out alive on the other end. And I don't know that anyone's looked to see for just intact dead cells. But my hypothesis would be those cells would be obliterated during the digestion, and that's where that lysate would come from. And we have done samples where everything I showed you, we were injecting that cell lysate IP. I did have data, I think in the paper, I didn't put it in the slide, where we gave the mouse oral cell lysate and it worked. So I think it's a digestion mechanism.
Okay. So before I go ahead, do you guys have anything?
I have one question. Do you think that freezing the breast milk somehow affects this, cell transferred immunity? Because many women just, freeze breast milk for comfort. So I was wondering about it.
Yeah, for our one specific thing that we're looking for outcome, I don't think that it would matter. Now, obviously, lots of proteins are more sensitive to freeze thaw than others. I mean, we're obviously intentionally freeze thawing it, and I know most women would only freeze it once and thaw it once. Now, whether or not that does anything to IgA content or any of the other potential beneficial factors, I have no idea. But for our work, a lot of the samples that we got ended up being frozen, right? Because shockingly, no woman wants to come into the hospital on day 4 postpartum. So they froze it and then brought it later on.
Right, thank you.
I have another general question, more not regarding this kind of like transfer of antibodies or some IgGs, but during the pregnancy, and mostly at least to my knowledge, I think, is it IgG which is transferred from the mom to the baby through the amniotic fluids, but placenta. And do you think any of these IgGs, if they were developed through let's say some sort of vaccinations or some sort of already suffered disease from the mother, will that baby have some kind of immunity against these particular things? Like let's say in these situations, the mom is vaccinated with a COVID vaccine. I mean, I'm not completely knowledgeable about what exactly is the antibody formed in this, but for example, if it's an IgG and this is transferred to the babies, will the resultant baby also have some kind of immunization?
So yes and no. So there is actually already one case report of a baby who had COVID antibodies, who obviously got them from a mom who was vaccinated. Whether that's at a titer that's protective is going to be the question. So when you're asking, well, will that, so yeah, if the mom transferred enough of it, you would anticipate that there would be protection. The other thing is that the mom should have substantial amount of IgA to whatever she was vaccinated against. And so when the breastfeeding happens, you'd expect that to team up. But yeah, if there was enough of it that could cross the protective titer threshold, sure, they should be born immune to that.
Yeah, so that means that if they're born like with IgGs and again they get their IgAs, it'll be like a double vaccination.
Yeah, well, keep in mind that newborns don't make any of their own antibodies for months. And so they need mom to supplement that from the placenta, right? So even if the mom's not breastfeeding, the baby's IgG in its system for the first 4 or 5 months is all mom. So you're going to need that no matter what. So yes, I think now the question is always, well, how protective is that in today's modern environment where your baby gets sick, you can take it immediately to a hospital. Obviously, certain parts of the world, that's not the same conversation. So I think evolutionarily, it certainly would not have been. So yes, I think those are vital. And so I think this might represent a mechanism for passive cellular immunization for kind of cellular functions that could protect that baby for those first few months during breastfeeding. I didn't get into it, but there is also evidence that the placenta itself becomes overly enriched in CD8 T cells. And they know that a bunch are coming in, but they can't find where they're going. So you can label them with some kind of tracker dye. You'll watch them go into the placenta. They're not going whole into the offspring. So that now raises in my head, I'm biased, but it raises the possibility that those cells are being broken down by the placenta and the lysate is being shoved into the amniotic fluid or into the child's circulatory system the same way as the antibodies. So there's evidence for, again, every time you start looking at the immunology of early pregnancy and early postpartum, CD8 cells just keep coming back up, and it's hard for me to keep ignoring it.
I have a very wild hypothesis that I will not shy away at all from sharing.
Believe me, this is the place to come. If you're talking, I told you, transfer factor is still a very controversial topic. Go ahead.
So the way T cell receptors are selected in the thymus, T cell receptors could act like B cell receptors, but the only reason they don't is because T cell receptors are all selected to have moderate binding to the MHC as well as another peptide. So I wonder if the T cell receptor extract is kind of enriching for some very high affinity peptides that are derived from the pathogen, and it's bringing it closer. So the dendritic cell, what it's receiving is not just a T cell receptor, but another peptide, which has to be very high affinity since there is no MHC, to get in contact with the dendritic cell.
Yeah. I mean, in my head, in my image of the cartoon, I got the MHC1 out there, whatever that target antigen is loaded in there. On the other end, you could just have the hypervariable region that binds that. And under normal circumstances, right, the entire apparatus binds all around it. You'd have to have, like you said, high affinity to stick all by itself. Mm-hmm. And then you almost certainly have to have some kind of coupling factor, right? Whether it's, how it gets put together, I don't know, but something else has to be tagged on to that hypervariable region that then is inducing a basic IL-6 response out of the dendritic cell. And then those two things together, or 3 or 4, who knows, could create this outcome.
Because if that is true, then we could have, it's no longer a passive immune transfer. It could be an active immunity, because the recipient will have its own clonal expansion, and now they will be able to do a memory response, unlike antibody transfer, where as soon as the antibodies are degraded or they just die out, the response ends.
So you're saying an active.
So this
why would it have a clonal expansion out of the dendritic cells, you mean? Or...
because the dendritic cells are also then inducing the CD8s, right?
Right.
In the recipient.
Oh, I see what you're saying. Yeah. So if the host had any CD8 cell around, mm-hmm. That cross, yeah. I'm down with your crazy hypothesis.
This could be a vaccination strategy, maybe sometime in future, for pathogens that are more likely to be killed by CD8s than antibodies.
Yeah, we've talked about that. Again, it's hard. It's a heavy lift because no one wants to extract it out of a cell lysate, right? Mm-hmm. too variable, too many risk as to what you could also cross-contaminate with. If we can clone it, the theory would be, if I'm right, and I reserve the right to be wrong, but if we're right and the hypervariable region is what is driving the antigen specificity, and then some other tag along is driving the immune response, and all that you get is just a shift between one antigen specificity and another is just a difference of that peptide sequence of the hypervariable region, you could start to then churn that out, right? You could make a mix that had hypervariable region for OVA, and then one that had hypervariable region for COVID, and one that had hypervariable region for staph. And as long as you knew what to tag onto it and how they were assembled, you could just churn this out. Again, yes, as a vaccination strategy, as a treatment strategy as well. I would say that in the old work, when they did those injections in humans and then would do the skin test, if you do a one-time injection and one-time treatment, that skin test positivity will disappear at about 4 to 6 months. So it's not a vaccine the way you think of it with, varicella, where you have lifelong protection or something like that. It's not going to do that. But if you wanted to have, something that you're going to go into an endemic region, or as a treatment strategy for something that's otherwise extremely antibiotic resistant, some kind of pathogen, it's kind of bacteria in that case, right? So you could do something like that.
Okay. We have a question from the audience. What do you think is responsible for the residual activation of cells when depleting CD8 T cells?
Well, I mean, there's always going to be some you call danger-associated molecular patterns in there. If you just take a lysate, one of the arguments around, for example, lupus, is that these cells are not supposed to be exposed to internal cellular components. You're not supposed to be awash in DNA, and maybe that slowly induces some kind of DNA responsiveness. So, if you just take the lysates, there should be some dirtiness to any of those assays, which is why, again, another reason why this is a heavy lift to convince people until you can purify something out and then say definitively, hey, look, this is what we found.
Okay. Just last one for me. When I'm donating plasma, could I be transferring these lysates to somebody?
No, this does not. These cells, when you take back in the day, right? They took the CD8 cells, cultured them profusely, and took the supernatant off or something like that. You take serum off, it is not in there. So people would subsequently discover like cytokines, right? So you stimulate the CD8 cells, they may be spitting out IL-17 or interferon gamma, and you can pick that up. But these cells do not just naturally release that. So, which is, so again, another confusing situation, but it would make sense if you're if you're putting the CD8 cell into the stomach of the offspring, now all bets are off. Those things are going to get broken up into pieces. So I do think that there is a natural self-assembly that must be going on. Because, if you had, for example, if you had the TCR, we don't know what the protease cleavage sites are, but if the natural gut proteases cleaved at the right spot, right? And if you had evolution to co-develop those at the same time, you'd cleave at the right spot to take out the chunk of the TCR you wanted, and some other cellular component would have to just naturally bind to it. Again, that's a lot of hand waving and a lot of conjecture, but that's what the mass spec is for. But no, there's no evidence for secretion. And when you do a regular blood transfusion, they should be taking the white cells out. If they don't, and they just do white cell irradiation, you might transfer that, but that would depend on whether or not it got broken up enough when it got injected into the individual.
This is so cool because it opens up an area of research where the protease cocktail in a newborn's stomach could have the effect on the immune system.
I mean, there's got to be a reason it's so tightly regulated in terms of every, dormice and blue whales have a CD8 enrichment. That's crazy.
We have one more question from the audience. Do you think that the DAMPs are necessary as an adjuvant for your CD8-dependent response, or do you think that the isolation of the hypervariable region would produce sufficient immunity?
I think the hypervariable region by itself, no way. Was that going to do it? The question is kind of twofold. Like, is there something else in the mix that has to come along with it, versus is there something in the mix that's tagged to the hypervariable region? So is there something is it a bound organism that I in theory could pull out in one unit, or would I need that other kind of quote unquote cellular contamination? Because to be fair, right, if any of this is correct and it's happening under natural feeding circumstances, there would never be a world in which the offspring got an isolated, purified transfer factor protein, whatever you wanted to call it, right? It would always have to have that. So that's a fair question. I don't know. I guess if we can genuinely clone out the differences between an OT1 and an OT2, if it doesn't have an effect in vitro or doesn't have as much effect in vitro, then yes, we're going to have to start talking about other things. We did find a role for TLR4, which would argue some LPS, or for humans, it could just be saturated fat production out of the milk can actually stimulate TLR4. Whether those two things are effective or not, I don't know.
Okay, thanks a lot. I'm sorry, my cat is very much interested in this talk. And
no problem. I mean, just the mammals hanging out right now.
Okay. So do you guys have anything else?
Yeah, I have one more question quickly, because they always kept saying that drinking cow's milk as a child was beneficial because calcium, vitamin D, and so on and so on. And would it be beneficial because of this transferred immunity to some cow pathogens, or not really?
Maybe. So the companies that I said at the very beginning, I thought, these people are charlatans. They're just hawking some dietary supplement. They sell colostrum. And I've seen their facilities. It is colostrum. It's not like just a bag of sawdust that they're passing off as colostrum. There's reason to think that it might be in there. Now, of course, you have to start. Now you get into like, all right, well, does this particular cow have more CD8 versus CD4 responsiveness? And is that milk that you got from the grocery store from a cow that has been forcibly pregnant, right? Dragged out the feeding for like 5 years. They've been milking for 5 years, in which case you don't expect the same ratios versus those earlier feeds. But yeah. Now, the other thing to say is whatever the cow is trying to pass on is to protect the baby cow, not you. So whatever pathogens you got to worry about is probably a different world. But yeah, I would think if we're showing evidence between crisscross between mice and humans, I don't think it's far-fetched to think that there's something in the cow milk that might have a similar transfer immunity. Whether it's relevant to you or not, I don't know. They did do a study. One more thing, they didn't do it for the milk, but at the very early stages of this, they would actively immunize the cow to something. And then they would take the cow's splenocytes and could show you a transfer between cow and human, showing that worked. Again, they didn't think to look at the breast milk. They wouldn't have known to, but we don't know.
If this was known during Edward Jenner's time, I'm sure he would try to give a cowpox infected cow's milk to a human, and that could theoretically work.
I like it. Let's go find some cowpox infected cows.
Yeah, because the ones we are getting milk from, I'm sure they are kept in somewhat hygienic conditions. So they should not be infested with diseases.
I like it. I'm sure the EPA will not, but I'm down with it.
If it's for science, it's all justified.
Right.
Every superhero villain ever.
Right. There you go.
Okay, guys. Do you guys have any other questions?
Yeah, I do. I have a question. Just like more like a general question, and I know about the innate immunity that breast milk transfers to infants from parents, from the mom. So how about in case of babies that take formulas? Is there like any difference in like the cellular immunity, or is there like anything we should be concerned about?
Yeah, concerned. I don't know that I'd use that word, because again, depends on where you are in the world. But I will say the parts of the world that are predominated by the possibility of bottle feeding are unlikely to be the ones that have a huge amount of background disease burden, right? So like rural Nigeria isn't bottle feeding at a high frequency versus Baltimore, right? So the places that you're going to have some of those endemic, truly dangerous pathogens still floating around are unlikely to be bottle feeding. So when you say, is it important? It's a question of like, does a mom in New York City have to panic if she's not able to produce milk and she's going to worry her kid's not going to be fully immunized? I would say no to that. But in terms of bottle feeding and breastfeeding, there's that one study that I showed you, that's from the 70s, that said that you could pass on the tuberculin reactivity in the baby cells. There's a gazillion studies that would show that breastfeeding is protective against various infections and against all the other things in the world, but that they're not sorting out the cellular fraction of that. So I think it would be hard to know. But there is 0% chance that bottle-fed infants are getting whatever this component is. There's no cellular byproduct in the formula. So again, if you could find it and you could begin to synthesize it in a safe way, and depending on how much antigen co-stimulation you needed, you could, in theory, start folding it in to bottle feeding in a way that would protect the offspring to those things at which the mom is more frequently exposed. And so I guess to take one step back, I told you that the immunization discrepancy between multiple shots in the guinea pigs versus one in the mouse. So when you float the antigen in CFA, it's basically a Depo-Provera shot, right? Everyone knows Depo for the birth control pills, right? Birth control is a one-time injection every month, right? And so you do that injection in the mouse, it's floating in oil, and so the immune system is kind of low-grade exposed to that the whole time while it's sitting there, versus floating it in an aqueous solution as what we're used to for a normal vaccine. Why might that matter? So that the differences between the central memory and the effector memory cells. Why this happens, I don't know, but things that you have low-grade chronic exposure to will tend to induce effector memory responses. Things that you have very intermittent massive exposures to will generate central memory. And so that might be another thing. So if a mom were going to want to preference passing on protection, the smartest things to do would be things that you're going to be exposed to. So low-grade, chronic, ubiquitous pathogens in your environment, pass on the immunity to that, versus things that you only see once every decade, that you don't want to gamble the kid actually is going to need that. So that's what I think is happening. I think the mom is passing on kind of the things that are low grade in her environment. And so you could figure out what those were in breast milk and then backfill that into formula in ways that is just simply not there right now. But again, whether that matters in most industrialized countries right now, I don't know. I don't think this is the cure for rotavirus or something in the kids, but I think you could use it more targeted to things, like you said, like a vaccine, a prophylactic, things that don't have good treatments, that kind of stuff.
Yeah, I think this would be a very good time to wrap up the discussion. Thanks a lot, Ian, for coming here today. I think without the Journal Club, I wouldn't know this much of a history behind this work.
Yeah.
And it was very, very informative session. I'm sure the audience also enjoyed it. And I personally learned a lot today. And I'm going to tell some of my friends the benefits of cellular immunity in the breast milk.
Go for it. Spread the word. Fight back against the dogma of the old days.
Yeah. And everybody watching us today, the video will be there on Facebook and YouTube for watching later. You guys can watch it whenever you like. Thanks a lot for joining today as the audience, and thanks a lot, Ian, for being here. And yeah, wish you good luck for more research. This paper has given rise to more questions, a lot more questions. Right.
All right, I'm gonna pick all you guys as reviewers for when I get the mass spec signature pulled out.
Oh, definitely, 10 out of 10, 100% recommended for publication.
Thank you all. Wish you guys the best.
Thanks a lot, and everybody joining us. See you guys in 2 weeks for the Journal Club number 20, and see you then. Bye bye.